Alright, good afternoon everyone. My name is Bunhua, marketing specialist from GenScript, and we are the world's leading biotech company with gene synthesis, protein, antibody, and preclinical drug development service capabilities. In today's webinar, we are going to discuss how can we enhance therapeutic discoveries with peptide libraries. So let me introduce our speaker today, Dr. Hao-Wing Leong. She is the field application scientist at GenScript who specializes in protein expressions and antibody production services. Dr. Leong graduated from the National University of Singapore in 2011, majoring in pharmacology and neuroscience, and worked as a research fellow in Duke and US Medical School thereafter. Dr. Leong has more than 10 years of research experience in molecular biology techniques and extensive knowledge in protein structure modeling, protein expressions, and its purification. This webinar will take around 45 minutes, and it will be a recorded session. You can type in your questions in the Q&A box, and we will answer them after the presentations. If we didn't get to answer your questions during the Q&A sessions, we will be sure to take down and email back to you. Now, over to you, Dr. Leong.
Yep, thank you, Bunhua, for the kind introduction. Good afternoon, everyone here. I'm Hao-Wing, and I'm the field application scientist from GenScript, working on the peptide portfolio. So thanks for taking time off to come for the talk on boosting your therapeutic discoveries with peptide libraries. So for today's talk, I'll be first talking about the common bottlenecks observed in many therapeutic discoveries, followed by what is a peptide library and what are some of the applications that require peptide library. The different types of peptide libraries and some case studies on how peptide libraries are used in the different applications, in particular for therapeutic discoveries. I will also touch on the importance in the quality of the peptides for us to progress with our therapeutic discoveries. So in many of the therapeutic discoveries, the identification of the targets is crucial. And many of these targets are protein in nature, with its distinct secondary structure, tertiary structure, and even quaternary structure. So such 3D structure often results in the hiding of important sequences that are important for the function of the proteins, for example, to elicit the downstream signaling to effect a therapeutic response. In addition, due to the 3D structure, such important sequences could be spanning over multiple epitopes, making the identification of crucial therapeutic sequences difficult, or the creation of antigenic sequences difficult. Excuse me. Considering such bottleneck and the limitations, so peptide become a possible solutions for probing into the mechanism of action, and also act as a simple means of targeting antigenic sequences spanning several epitopes. And this is because peptides are synthetically accessible, and they are easily amenable to chemical tailoring. They are also be able to bind to shallow surfaces, which are unlike other chemical entities. And not only so, they can also be non-canonical amino acid incorporation to enable changes in the stability or the peptide structure. Such changes often enables them to be more proteolytically stable and even more cell penetrant. So in addition, they are easily amenable through chemical means, and thus they will be easier to increase the binding affinity just by simple modifications. So as such, to speed up the therapeutic discoveries, the identification of the targets and such epitopes is paramount, and peptides could aid in this. Having a peptide library, which is a presentation of multiple short linear peptide fragments, would enable such target identification. So typically, upon identifying a bioactive region, the precise epitope targets could then be determined through a combination of amino acid sequences, mutations or not, depending on the issues in questions. So this I'll be illustrating on in later slides on how it could be done. So peptide sequences are then presented in their appropriate assays for target identification or even function determination. So there are two major ways of creating such peptide libraries. And one is through a phage display, where the peptides are expressed on the surface of these phages upon the transfer of related DNA into these phages. So specific clones are then selected based on the binding affinity to the target, thereby determining crucial epitope information. So one such advantages of such peptide libraries are that they are simple and are not limited by peptide length. However, such determination of binding affinity is a labor-intensive task and are limited to natural L amino acids. However, in a synthetic peptide library, peptides are synthesized chemically on a solid phase or liquid phase, depending on the length of peptides in question and also the quantity of peptides required. The chemical synthesis allows for flexibility and also the option of adding the amino acids. In addition, they require actually less time. So in some cases, the usage of the amino acids are desirable as it could stabilize the peptide as they are more resistant from proteases. So as briefly mentioned, peptide libraries are good with identifying target-specific epitopes. For example, if you have a protein target and would like to find out the therapeutic, if the therapeutic antibodies could bind specifically to your protein target antigen, or if you would like to optimize the binding affinity of the antibodies towards the antigen, the identification of the epitope or epitope mapping is crucial. So to do this, we can break up the antigen into smaller peptide sequences in a systematic manner, forming a peptide library. So the therapeutic antibody drug lead would then be allowed to bind with the peptide libraries. The epitope could then be identified based on binding affinity. The same could be applied for T-cell epitope mapping. So instead of antibodies, MHC molecules could then be used for this application. It could also be used for in cancer cell biomarker. So for example, a random series of peptides could be chemically synthesized, forming the peptide library. And specific protein of interest, for example, the receptor protein specific for a particular cell type or biomarkers for cancer cells could then be expressed and purified and added to the peptide library, thereby identifying potential peptide drug therapies specific for the type of cancer. So in addition, identifying such biomarker binding peptides could aid in gene therapy in cancer treatment, which is currently quite hot right now. So the transfer of gene example, like for example, the siRNA for gene therapy treatment has always been a bottleneck because of the inherent anionic properties of the RNA molecules, rendering them difficult to pass through the cell membrane. So conjugating peptides with specific binding to cancer biomarkers not only target its siRNA specifically towards the cancer cells, but also aids in the cell penetration with the cell penetrating properties of the peptides. So as such, peptide libraries are very useful in various diseases, such as drug discovery, such as T-cell immunotherapy, vaccine development, antibody therapy, and also in cancer research. So other use for peptide libraries are in peptide bioassays. The peptide libraries could be presented to embryonic stem cells, for example, to understand the mechanism of stem cell differentiation, and triggers for stem cell differentiation. It could also be used in disease diagnostics. So the assay is similar to that of the cancer biomarkers epitope identification. So it could be used for optimizing drugs for improved bacterial activity, antibacterial activity. So peptide libraries could be presented to the microbes and to identify the best peptide sequence that could elicit antibacterial activity. So considering the many applications for peptide libraries, how can we generate or design a peptide library? So most importantly, we need to identify the bioactive region of interest. And first, we also have to define our question and choose the appropriate library. So in GenScript, we have free online tools for different kinds of libraries to probe and answer different questions. So upon choosing it, all one needs to do is to input the sequence, and the peptide libraries will be generated with just a click. So thus choosing a peptide library that is appropriate for the scientific question to be answered or the applications is crucial. So in GenScript, we have 7 online library design tools, which I'll be illustrating in the following slides on the type of applications each is good for. So for example, for an overlapping peptide library, it is typically used for linear or continuous epitope mapping. It is a series of short peptides overlapping each other, and the length of the peptide will be set according to how fine and the resolution one required to define the epitope. But typically, it ranges between 8 to 20 amino acids, and the offset, that is the amino acid gap before the start of the next amino acid, next peptide, will preferably be 2 to 4 amino acids. And so the smaller the offset, the higher resolution of the peptide, and such overlapping peptide, which is in epitope mapping, is good for applications in determining the antigenic sequence for epitopes in T cells or in antibodies. Such library is also good for screening substrate sequence for enzymatic proteins or to determine the binding sites of protein receptors. So in this study by John and all, published in Vaccine, they have actually generated such overlapping peptide libraries with us, with GenScript, to develop a better mRNA vaccine targeting the immunodominant CMV T cell antigen PP65. So against the CMV. So studies have previously shown that the CMV glycoprotein, the GB, and also the pentameric complex are candidates for vaccines. And in this study, they wanted to determine whether additional of the vaccine expressing the PP65 antigen could actually improve this mRNA vaccine. So 1 week post-vaccine boost, the splenocytes from the indicated groups were stimulated with either pooled PP65 peptide libraries and polyfunctional IFA gamma T cell responses were measured by ICS and analyzed by flow cytometry, allowing them to determine that PP65 could actually elicit is on its own the T cell response, validating the success of PP65. In addition, the observation of additional 3 different antigen together and the observation that there seems to be an inhibition in the overall T cell response allow them to determine the mechanism of action of the vaccine and identifying that there is some sharing of some epitopes between PC65, PC and PP65. So this push the researchers to develop a vaccine technology, a vaccine strategy as shown here, with the addition of PP65 at different times to boost the vaccine response. So next, let's take a look at another type of peptide library, that is the alanine scanning peptide library. So such library are typically used to identify key amino acid for binding function, for protein stability, or for when researchers would like to identify which amino acid is crucial for the conformation of protein structure. So typically, such input of the sequence would require about 6 to 200 amino acids, and the alanine scanning library algorithm will systematically generate and mutate one residue at a time to an alanine. And this allows us to identify key residues in the epitope and to locate binding sites of the protein, and also to evaluate the function of the protein or the enzyme. So in this case study, in the next case study that we perform in-house in GenScript, we would like to identify the key residues in the CDR variable region of the SARS-CoV-2 antibody. So to do that, we have actually generated an array alanine scanning library to replace all non-alanine residues in the CDR region to a serine to probe into the function of the native alanine residues in the CDR region. So the native alanine is also replaced to a serine to probe the function of these alanine residues in the CDR region. So the red dots shown in the figure show crucial residues that are important for the CDR for the antibody antigen binding. Upon identifying such important residues, we could then optimize the binding of the antibody through mutations of these residues. So now, such optimization of antibody binding could be performed using the positional scanning peptide library. So such peptides are good because they are able to allow for peptide sequence optimization. So for a particular position, possibly identified through the previous previous library, like the alanine scanning peptide library, this position then can be substituted with all other 19 amino acids to identify and optimize the peptide binding, increase the activity of enzyme substrates, or to enhance the antibody or T-cell epitopes. So in the following case from Avantgarde et al., published in POS 1, they had actually generated a positional scanning library with GenScript at all the residues at the tab in the Cap18 sequence to obtain an enhanced Cap18 variant that is effective against different bacterial species. So Cap18 is actually an antimicrobial peptide. They are alternatives to classical antibiotics. So in this figure, the original peptide sequence is shown above here, and the respective substitution for each amino acid is shown here. The number indicates the minimum inhibitory concentration to each mutant upon substitution to that amino acid. So for example, the red here will indicate a higher MIC that is mutating this residue to the respective residue here would decrease its effectiveness of Cap18, indicating that this could be important residues. And those with a small value of less than 1 would then indicate, let me see, like less than 1, would indicate such residues could be good to optimize therapeutic function of Cap18. As such, on the right here, the researchers had indicated with the following diagram the important residues and how, for the different activities, and how they could be mutated to what kind of amino acid to enhance the function of Cap18 as an antibiotic development. So another way that we could optimize the peptide sequence could be through a random peptide library. So unlike the positional scanning peptide library, so the random peptide library uses a shotgun approach, resulting in the randomness of the peptide library. So input sequences could be between 10 to 200 amino acids, and they are good for peptide sequence optimization, but also in target identification and drug discovery. So in this case study from Quantararo et al. Published in Nature Communications, they have used a random library by fixing the serine, phosphorserine to be specific, in the middle and flanking it with 4 other ferric amino acids on either side. This gives them a big peptide library of to the power of 10 to the power of 8 members, allowing them to probe and identify the consensus sequence for 14-3-3 gamma binding. As some of you may know, 14-3-3 is a hub protein which interacts with a range of disease-relevant proteins such as P53, RAF kinases, estrogen receptor. So these researchers in this study would like to develop modulations of such interaction through phosphorylation. And as such, with chemical synthesis of the peptide libraries, the addition of a non-canonical amino acid is easy. And this demonstrates how peptide libraries are crucial in actually developing therapeutic modulators. And of course, sometimes we need to have some kind of controls with regards to the design of the peptide library. And Scrambled peptide library will be good for obtaining such information. What is it used for? Usually the Scrambled peptide library, it provides us with the highest variation in terms of the peptide. As such, it is actually good to specify the library size for such screening. And this, as mentioned, could act as a negative control, but also will be good for probing molecules of interest, antibodies, and DNA. So an example of the Scrambled peptide library is demonstrated by Raghav et al., published in the International Journal of Nanomedicine. So they had used the design tool in GenScript to create 80,000 peptides with 50 peptides in one library of input length at 13. So to identify synthetic epitopes that could bind to the GATA1 and to regulate the myelopoiesis. They would like to find out that the synthetic peptide analog would block the interaction between GATA1 and PU1 to allow the interaction of PU1 with Seagen to promote myelopoiesis. So as shown here from the libraries, they've eventually identified 3 peptides that have such potential with higher binding affinity to GATA1 and lower with Seagen compared to the control. So from an original protein sequence, we could also generate a truncation peptide library to probe into the minimum length that is required for activity. Usually this is upon some information that is obtained through adenine scanning library studies, where we have already identified some crucial residues in the epitope, but would like to map the minimal binding site for core protein binding. So here I have presented another case study in which the Nagans and all published in Antimicrobial Agents and Chemotherapy illustrate how such truncation libraries are generated with the sequential deletion of hexapeptides from either from the N terminal or the C terminal or from both ends of the native peptide. This allows them to decipher crucial sequences that are required for antimicrobial properties with reduced toxicity. As shown in this graph, they had identified 3 peptides, the LL37 to 37, LL13 to 37, and LL19 with lower toxicity and indicated the decrease, as shown by the decrease in the release of LDH. And they have also improved microbial properties of the peptides, as shown by the PI staining. So this truncated peptide library could also be used to identify T-cell epitope. So optimal conditions for this kind of library are short peptides sequences from 8 to 11 amino acids on the MHC1 complex or 15 to 24 amino acids on the MHC2 complex. So applications include testing all possible T-cell epitopes across the protein of interest. So to better illustrate on how the T-cell truncated peptide library can be used, I have presented the study by Yoshihiko and all in a Journal of Immunology, whereby they demonstrate how T-cell truncated library is used to deduce the binding epitope of the T-cell. So first, using a T-cell truncated peptide library to identify the peptide region, epitope region, they then subsequently use different peptide libraries such as the random library here and also the positional scanning library here on a number of residues to identify crucial residues to enhance activation of the T-cell. So I have demonstrated the various peptide libraries for different therapeutic discoveries, investigations, and also its optimization. Some of which are T-cell immunotherapy for vaccine development, for cancer research, antibody or T-cell epitope mapping, and even antimicrobial drug discovery using different library design. So the table here give a brief summary on the possibility of the different libraries for each of these applications. And of course, depending on the applications, the purity of the peptides required will be different. For example, in biomarker discovery or epitope mapping, or there's some T-cell binding assay, usually crude purity will be sufficient. However, in more cells sensitive assays such as immune monitoring or cell-based assay, whereby just a little bit of impurity would actually show different kind of results on the cells. Such would require, higher purity of the peptides or of the peptide libraries. For example, especially when you need to quantify, quantitative assays will again need a higher purity of maybe more than 80%. And of course, in drug and vaccine development, and also in preclinical trials or sensitive assays such as NMR, at least 90 or even 98% of purity will be required. So in GenScript, in additional to synthesizing peptides, just normally we do have, peptide libraries available when the number of peptides required by the client is more than 24. So for a crude peptide library, they are good for bigger amount, up to 20 milligram. And for clients whose applications do not require that high of a purity. We also provide purified peptide library package where client can select between a range of purity from 70 to 98%. We can also provide such libraries for quantity up to 9 mg. All peptides will be provided with HPLC and MSQC and its related documents. And in addition to that, there is also a comprehensive list of modifications available for such peptides, such as biotin conjugation or fluorescent dye conjugation or phosphorylated amino acid, methylated amino acids, or even incorporation of unnatural amino acids. So this slide shows the bioinformatics tool that we have in GenScript for generating the peptide libraries. So simply one just have to input the protein of interest and set the parameters here. For example, here, this is for the overlapping peptide library. And then with just a click, the peptides corresponding to the condition set and also the library selected, the peptides will be provided. So in addition, in GenScript, we also have different bioinformatics tools to allow us to understand the characteristics of our peptides. For example, is isoelectric points or hydrophobicity, or also what are the difficulties in synthesizing a particular peptide. So we have at least 15 years of experience generating peptides for our clients, and we have in place the PEP HTS, we call it the high throughput system, library platform to ensure the delivery of various libraries for our big pools of clients. So on this platform, we can produce in a high throughput manner at 18,000 peptides per month, so that we can ensure fast turnaround time as it operates 24 hours and is fully automated. So we also have a series of Immunopep QC available to ensure the quality of the peptides generated. We are ISO 9001 certified total quality management TQM platforms, ensures that each custom peptide is triple checked for quality via both mass spec, MS, and also HPLC analysis after each step during the peptide purification, and also is QC procedures. So in addition to such capabilities, we understand that many of these peptide libraries usage could be for T-cell immunotherapy or cancer therapy. So GenScript also has formed a collaboration with Cayman to perform immunopeptide profiling, which is a deep sequencing analysis of the MHC-associated peptides to identify new antigens or cancer biomarkers and potential immunogenic sequences from MHC1 or 2. So human or mouse tumor cells are lying under optimal conditions for releasing the MHC complexes. And using the amino affinity chromatography, MHC-specific antibodies enrich the pool of MHC class 1 and class 2 from the lysates. And MHC-associated peptides are then released under gentle eluting conditions and separated from the captured MHC molecules. Then these peptides are then sequenced by LC-MS, MS, and then matched to the protein sequence database or RNA sequencing data, as well as analyzing for mutations or post-translational modifications to facilitate the identification of potential neoantigens. And of course, sometimes, the synthesis of such neoantigen peptides can sometimes be difficult because neoantigens can range between 8 to 50 amino acids, and peptides that are more than 20 amino acids are more challenging to synthesize. And at times, the neoantigen peptide selected may also have a neutral charge, making purification of such peptides more difficult and also is in synthesis more challenging. So hydrophobic residues peptides are also notorious for synthesis due to its insolubility in water, making purification difficult. And in addition, these hydrophobic peptides may tend to aggregate too. So in view of this, GenScript has been constantly developing new technologies and algorithms to aid in the synthesis of difficult peptides. We have the new algorithm for us to determine how difficult a peptide is to be synthesized based on its length, aggregation properties, and also the hydrophobicity and the isoelectric point. And upon this, the scientists in GenScript then could determine the type of synthesis that be more suitable for such peptides. We also have, different processes such as Hysin process, which is specifically developed to synthesize difficult peptides. So GenScript has rigorously optimized the reaction temperature, condensation reagents to increase the coupling efficiency. This allows us to have a better success rate of 91%, even for hydrophobic peptides. And we are also the first CRO to have double coupling technology, in which we have used the coupling reagents from Hysin and applied with the microwave technology, in which we have optimized the reaction conditions for this purpose. And so we are able to increase our success rate, synthesizing previously unable to synthesize peptides at a high purity. So with this as any note on our capabilities of synthesizing good quality peptides, I would like to thank you for your attention and time. Yeah, thank you. Any questions?
Yeah, thank you so much, Dr. Leong, for your wonderful presentations. To all the attendees, you may ask questions by typing your question in the Q&A box. Okay, hold on. I think we have a few questions, so let me read it out for you. Okay, we have one question here. When designing overlapping peptide libraries, what are some of the precautions that we have to take note?
Okay, for such libraries, so as mentioned before, the bigger of the offset, the higher chance that we will miss some important amino acid combination. So choosing a shorter offset number, so typically this should be about, if possible, 1/3 of the desired peptide length will help us to ensure that the libraries, cover the required epitope probing. So yeah, the offset number.
Yes, thank you. Okay, the next question is, what can I do if I have a library with many hydrophobic residues?
Yeah. So there will be times, when, the protein sequence that we are working on will generate some hydrophobic peptides, that are, difficult to purify and solubilize. So in GenScript, we also have, the peptide calculator to probe into the characteristics of the peptides I mentioned earlier in my slides, to allow us, beforehand, before synthesizing, to allow us to have a better understanding on the properties of the peptides and how easy it is to synthesize. So if the peptide libraries have many hydrophobic peptides, one could consider either choosing a lower purity, because usually with a lower purity, yeah, it'll be easier. And then, or you can also substitute, some of the non-crucial hydrophobic residues to others, such as glutamic acid or aspartic acid or even lysine. Or sometimes if it is too difficult, maybe if the application permits, then we may need to choose, different peptide sequences. Yeah.
Okay, what is the peptide purity that you are referring in this webinar?
Oh, okay. So the peptide purity is actually the amount of correct peptide relative to the total analyzed, through the HPLC that is absorbed at 214 nanometer. So this it may include deleted or truncated or incompletely deprotected sequences. So some other impurities also include like TFA or some salt or water or solvent. So GenScript actually can provide peptides with a different purity in that sense. Crude 70, 75, 80, 85, 90, 95, 98 based on the applications that is required. Depending on the client needs. So to purify peptides, usually we use reverse phase and also ion exchange HPLC. So and usually ion exchange HPLC is used for the purification of longer peptides or larger quantity of peptides. Yeah, so this is the purity that we are referring to, and it's quite crucial in some assay. So yeah.
Yes, thank you. The next question would be, what kind of modification services that you provide?
Okay. Yeah, we have various modifications. So at least 300 different modifications available, at least. So such modifications are usually good to improve the peptide stability, for example, such as to incorporate the amino acids or some head to tail cyclization or amidation at the C terminal. Or sometimes, modifications are required to enhance the immunogenicity for antibody development or production. So such modifications are also available, such as to conjugate to BSA or KLH or OVA. So in addition, we also have various fluorescent peptide modifications available, such as for FIT-C, for example.
Yeah, thank you. Okay, to all the attendees out there, we still have quite a lot of time to answer your questions. So if you have any questions regarding this webinar or regarding our peptide library services, you may feel free to type your question in the Q&A box. We'll answer them. Okay, we have one more question here. What are the guarantees for the peptide synthesis under GenScript?
So for our peptides, they are packed with the ion shield technology to eliminate experimental variation from peptide oxidation or sometimes delinquencies. So delinquencies. So for peptide synthesized under GenScript, so they will be guaranteed on the also not only that, there will be guarantee on the quantity. For example, if you have orders less than 20 mg under the regular express, they will be guaranteed on the quantity. So what else? So peptides in the peptide libraries can also be guaranteed on the QC, because we will provide with the HPLC and the MS report for each of the peptide that is synthesized. And also we also understood that the residual TFA in custom peptides sometimes may cause inexplicable discrepancies in assay data, especially in those cellular assays. So we also provide a guarantee TFA removal services. So if clients are opting for this service, they will be guaranteed that the TFA level is less than 1%. So these are some of the different guarantees that we have for the quality of the peptides that are synthesized under GenScript.
Okay, thank you. Okay, I see no questions from the floor, so I think we'll close this webinar. So thank you once again, Dr. Leong, for your great presentations, and thanks all of you who joined this webinar today. I hope you find this webinar useful and beneficial for your work. And thanks everyone, and we'll see you again. Have a nice day and goodbye.
Okay, bye-bye. Thank you.